Hello students, today we will learn about polysaccharides, polyhydroxyalkanoates and lipids. So let's begin. Microorganisms in the presence of excess supply of carbon are capable of producing storage compounds. These storage compounds are polysaccharides and polyhydroxyalkanoates for some microorganisms and lipids for some yeast and fungi. Now let's begin with polysaccharides. We'll start with polysaccharides. The microorganisms can produce large amount of polysaccharides in the presence of surplus carbon sources. Some of these polysaccharides, example glycogen, serve as storage compounds. The polysaccharides excreted by the cells referred to as exopolysaccharides are of commercial importance. The exopolysaccharides may be found in association with the cells or may remain in the medium. The microbial polysaccharides may be neutral. Example, dextran, sclerogans, or acidic. Acidic means xanthin and galen in nature. Acidic polysaccharides processing ionized groups such as carboxyl, which can function as polyelectrolytes, are commercially more important. Now let's know about the applications of microbial polysaccharides. Microbial polysaccharides have immense commercial importance. They are employed in the stabilization of foods and production of several industrial and pharmaceutical compounds. The commercial value of a polysaccharide is based on its ability to modify the flow characteristics of solution. Polysaccharides can increase the viscosity and are therefore useful as thickening and gelling agents. Microbial polysaccharides are of great importance in oil industry. By conventional methods, only 50% of the oil can be extracted and the rest of the either trapped in the rock or too viscous to be pumped out. It is now possible to recover such oils also by a technique called microbial enhanced oil recovery, in short MEOR. This can be done by injecting surfactants and viscosity decreasing biological agents, that is the microbial polysaccharides, example xanthan and emulsion. Now let's know about the production of microbial polysaccharides. The synthesis of polysaccharides favorably occurs in the excess supply of carbon substrate in the growth medium while limiting nitrogen supply. A carbon or nitrogen ratio of around 10:1 is considered to be favorable for optimal polysaccharide synthesis. The production process is mostly carried out by batch culture fermentation. By manipulating the nutrient supply, differential synthesis of polysaccharides can be achieved by limiting nitrogen supply in the medium. Mostly neutral polysaccharides are produced when metal ions are limited. Acidic polysaccharides are mainly synthesized. Molecular oxygen supply of around 90% saturation is ideal for good growth and polysaccharide synthesis. Now let's know about biosynthesis of polysaccharides. Microorganisms are capable of producing a large number of polysaccharides. The pathways for their biosynthesis are comparable to the processes that occur for the formation bacterial cell wall. It is estimated that there are well over 100 enzymatic reactions directly or indirectly involved in the synthesis of polysaccharides. Starting with glucose, appropriate sugars by transforming glucose to others are incorporated in the formation of polysaccharides. Now let's know about the recovery of polysaccharides. As the polysaccharide production increases, there occurs a marked increase in viscosity of the culture broth. The polysaccharides can be precipitated by salts, acids or organic solvents and recovered by employing appropriate techniques. Now let's know about microbial polysaccharides. versus plant polysaccharides. There is a lot of competition between microbial and plant polysaccharides for industrial applications. Production of plant polysaccharides is relatively cheap, although it is uncontrolled and occurs for a short period in a year. In contrast, production of microbial polysaccharides is well controlled and can be continued throughout the year. However, fermentation process for manufacture of cheap from plant sources polysaccharides is not advisable. Now let's know about general features of microbial polysaccharides. Of the several microbial polysaccharides, around 20 are of industrial importance. As already stated, the commercial value of a polysaccharide is mostly dependent on its rheological properties, that is, its ability to modify the flow characteristics of solutions. The microorganisms used for their production, their application, now we'll discuss. Now let's know about, first is xanthan. Let's know about xanthan. Xanthan, or more frequently referred to as xanthan gum, was the first polysaccharide available commercially. It is a well-studied and most widely used hypopolysaccharide. Now let's know about xanthan, chemistry of the xanthan. Xanthan has a molecular weight in the range of 2 to 15 into 10 to the power 4 daltons. The basic repeating unit of xanthan is a pentasaccharide containing glucose, mannose and glucuronic acid with acetate and pyruvate. We know. Basically, xanthan is a branched polymer with beta-linked glucose polymer backbone bound to a trisaccharide, that is MAN, GLCA MAN, side chain on alternate glucose residues. The mannose has either acetate or pyruvate groups. The number of acetate or pyruvate molecules in xanthan is variable and is dependent on the bacterial strain used. The culture conditions and the recovery processes also influence the quantities of pyruvate and acetate residues. It is believed that the viscosity of xanthan gum is influenced by the content of pyruvate and acetate. Now let's know about the applications. Xanthan gum is used as a food additive for the preparation of soft foods, ice, cream, cheese. It is also used in oil industry for enhancing oil recovery. Further, xanthan is used for the preparation of toothpaste and water-based paints. Now let's know about the biosynthesis. Biosynthesis, For the biosynthesis of xanthan, the monomers are bound to a carrier lipid molecule and then transferred to a growing polymer chain. The activated monosaccharide nucleotides, example uridine diphosphate glucose or UDP glucose, supply energy for the formation of glycosidic bonds between adjacent units. The biosynthesis of other exopolysaccharides is comparable with that of xanthan. Dextran synthesis, however, is much simpler, as we will discuss. Now let's know about the production. How it is produced. Xanthan is commercially produced by gram-negative bacterium Xanthomonas campestris. The culture medium usually consists of 4 to 5% carbohydrate, that is glucose, sucrose, cornstarch, hydrolysate, and 0.05 to 0.1% nitrogen, ammonium nitrate, urea, yeast extract and salts. The pH is maintained around 7.0 and the fermentation is carried out by batch culture for 2 to 3 days. Xanthan in the culture broth is precipitated by isoprene or methanol. These agents also kill the microorganisms. The precipitated xanthan can be dried and used for commercial purposes. Now let's know about genetic engineering of Xanthomonas campestris for xanthan production. The wild type X chemistries can efficiently utilize glucose, sucrose or starch as a carbon source. They are, however, unable to use lactose as a carbon substrate. Whey is a byproduct obtained in the manufacture of cheese. Disposal of large quantities of whey is a major problem in dairy industry. Fortunately, whey is rich in lactose, besides containing small quantities of proteins, vitamins and minerals. Attempts are made to use whey in fermentation industries. Genetically engineered eggs chemistries have been developed that can utilize lactose from whey for the production of xanthan. For this purpose, the E. coli laxi genes encoding the enzyme beta galactosidase and lactose permease respectively were cloned under the transcriptional control of X campestris bacteriophage promoter. This construct was first introduced into E. coli and then transferred to X campestris. The genetically engineered strains of X campestris expressed the genes and produced high quantities of the enzymes beta galactosidase and lactose permease. These new strains utilize lactose or whey very efficiently for the industrial production of xanthan. This is a good example of successfully converting a waste product, whey, into a commercially important and valuable product, a biopolymer, namely xanthan gum. Now let's know about dextran. Let's know about dextran. Chemically, dextran are glucans, polymers of glucose containing 1 to 6 glucosidic linkages. Some dextrans also have alpha 1 to 2 or alpha 1 to 3 and alpha 1 to 4 linkages. The molecular weights of dextrans are in the range of 15,000 to 5 lakh. Applications. Let's know about the applications of dextrans. Dextrans are used as blood plasma expanders for the prevention of thrombosis and in wound dressings. In addition, dextrans are useful in the laboratory analytical techniques for purification of biomolecules. Now let's know about the production process. Dextrans can be produced by a wide range of gram-positive and gram-negative bacteria. Example: leuconostoc, mesenteroids, and Streptococcus mutans. In contrast to other exopolysaccharides, which are synthesized within the cells, dextrans are produced by extracellular enzyme in the medium. The enzyme is dextransucrase, a transglucosidase which acts on sucrose and brings about polymerization of glucose residues and simultaneously liberates free fructose into the medium. The commercial production is carried out by using lactic acid bacterium L. mesenteroids by a batch fermentation process. Besides sucrose, the culture medium contains organic nitrogen source and inorganic phosphate. The crude dextran produced is precipitated by alcohol and then subjected to acid hydrolysis. In recent years, the alcohol precipitated polymeric dextran is subjected to enzymatic hydrolysis by using azo or endo-alpha dextranases to get dextrans of desired molecular weight. The resultant dextrans can be fractionated and dried. It is also possible to use a cell-free system for the production of dextrans. The Extracellular enzyme dextrasucrase can transform sucrose into dextran in a cell-free nutrient solution. This reaction is optimum at pH 5.0 to 5.5 and temperature 25 to 30 degrees centigrade. Now let's know about alginate. Alginate is a linear polymer composed of manuronic acid and glucuronic acid in a proportion ranging from 4:1 to 20:1. Some of the manuronic acid residues are acetylated and alginate is commercially produced by gram-negative bacteria Pseudomonas aeruginosa and Azobacter vinylandi. These are two bacteria. Okay, gram-negative bacteria. The type of organism used and the culture conditions determine the relative proportion of manuronic acid and glucuronic acid residues and the degree of isolation in alginate. The alginates with the high contents of manuronic acid are elastic in nature, while those with the high concentration of glucuronic acid are strong and brittle. Algal, that means seaweeds, alginates are also polymers of manuronic acid and glucuronic acid and comparable in structure with bacterial alginate. However, algal alginates lack acetylation. For commercial purposes, seaweed alginates are more commonly used than bacterial alginates. This is mainly because bacterial alginates are relatively unstable and get easily degraded. Alginates are useful as thickening agent in food industry and for immobilization of cells and enzymes. Now let's know about scleroglucan. Scleroglucan is a glucose polymer, that is glucomer, and it is a natural polysaccharide with beta 1 to 3 glucan backbone and single glucose residue branches, that is beta 1 to 6 linkage. The branching occurs at a regular sequence at very third glucose unit in the polymer backbone chain. Scleroglucan is a fungal hexapolysaccharide. It is commercially produced by Sclerotium glucanium, S. rolpsi and S. delphini. Scleroglucan is useful for stabilizing latex paints, painting links and drilling mods. Now let's know about Galan. Galan is a linear heteropolysaccharide. The repeating unit of Galan is composed of 2 glucose, 1 glucuronic acid and 1 rhamnose molecules. Galan is produced by Pseudomonas elodea. The acetylated Galan, which forms firm and brittle gels under the trade name Gelrite, has been developed by a reputed company in USA, Calco Inc. Galan is used in food industry even at a low concentration. It is a thickener. Now let's know about polyulan. Polyulan is an alpha glucose polymer, that is alpha glucan with alpha 1 to 4 and a few alpha 1 to 6 glucosidic bonds. Polyulan is produced by using the fungus Aureobasidium polyulans. It is estimated that about 70% of glucose is converted to polyulan during fermentation, although the time taken is rather long, 5 to 7 days. Polyulan is mainly used in food coating and packaging. Now let's know about codlan. Codlian is a beta-glucose polymer, that is beta-glucan. The glucose residues are held together by beta-1 to 3 glycosidic bonds. The exopolysaccharide codlian is commercially produced by employing alkali genes fecalis. Codlian like polysaccharides are also produced by other microorganisms such as Agrobacterium rhizogenes and Rhizobium trifoli. Codlian forms strong gels when heated to more than 55 degrees centigrade. Therefore, it is used as a gelling agent for cooking foods. In addition, codlian is also employed for immobilization of enzymes. Now, let's know about polyhydroxyalkanoates. Polyhydroxyalkanoates, in short PHA, are intracellular carbon and energy source compounds produced by many microorganisms. They are biodegradable polymers and are elastic in nature depending on the polymer composition. PHA are well suited for the synthesis of plastics, the biodegradable packaging materials. The conventional plastics made from coal or oil are not biodegradable. They survive hundreds of years and are major sources of environmental pollution, often resulting in ecological imbalance. A heavy demand for biodegradable plastic materials exists. There are some attempts to chemically synthesize biodegradable polyesters, polylactic acid and polyglycolic acid. The production of polyhydroxyalkanoates by fermentation is preferred for use as biodegradable plastics. Now let's know about polyhydroxyalkanoates. Polyhydroxyalkanoates, in short PHA, are intracellular carbon and energy storage compounds produced by many microorganisms. They are biodegradable polymers and are elastic in nature depending on the polymer composition. PHA are well suited for the synthesis of plastics, the biodegradable packaging materials. The conventional plastics made from coal or oil are not biodegradable. They survive hundreds of years and are a major source of environmental pollution, often resulting in ecological imbalances. A heavy demand for biodegradable plastic materials exists. There are some attempts to chemically synthesize biodegradable polyesters, example polylactic acids and polyglycolic acid. The production of polyhydroxyalkanoates by fermentation is preferred for use as biodegradable plastics. Now let's know about PHA chemistry and properties. PHA serve as lipid reserve materials in bacteria. Their function in bacteria is comparable to that of fats and oils in yeast and fungi. The granules of PHA stored within the cells are clearly visible under electron microscope. Some bacteria may accumulate huge quantities up to of a huge quantity of PHA, that is up to 80% of dry weight. Polyhydroxyalkanoates are linear polyester polymers composed of hydroxy acid monomers. The most commonly found monomers are 3 hydroxy acids with a carbon length ranging C3 to C14, carbon 3 to carbon 14. Now let's know about homopolymer PHA polyhydroxybutyrate. The most common PHA is polyhydroxybutyrate, which is frequently referred to as PHB. It is a polyester with 3-hydroxybutyrate as the repeating unit. PHB is the homopolymer PHA. PHB and as such is rather hard and inflexible. Being a high molecular weight compound, the accumulation of PHB is in huge quantities also does not significantly affect the osmotic pressure within the cells. The reserve carbon compound PHB can be oxidized to carbon dioxide and water releasing large amount of energy. Bacteria require energy although they are not growing to maintain pH gradient and concentration gradient of several compounds. This energy refers to as maintenance energy essential for the survival of the cells is met by the reserve material PHB. Now let's know about heteropolymers of PHA. Majority of polyhydroxyalkanoates, with the exception of PHB, contain two or more different monomers and are referred to as heteropolymers. These heteropolymers are usually composed of a random sequence of monomers and not different monomers in different chains. Besides 3 hydroxy acids, several other hydroxy acids are found in the structure of PHA. Example 4-hydroxybutyrate, that is 4-HB. In fact, it has been found that the bacteria are capable of incorporating more than 100 different hydroxylated monomers into PHA. This depends on the organism and the nature of the carbon source supplied during the accumulation of the polymer. The properties of PHA mostly depend on the nature of the monomers it contains. In general, PHA with longer side chains, example 3-hydroxy octanoate containing PHA and heteropolymeric, that is heteropolymeric PHA, are more flexible and soft. Now let's know about 3-hydroxybutyrate co 3-hydroxy valerate. The composition of the medium, the chemical construction of the PHA can be altered. Thus, when the medium contains glucose and propanoic acid, the organism Ralstonia eutropia produces a copolymer of 3-hydroxybutyrate and 3-hydroxyvalerate, that is PHB, as such as is hard and brittle, but the presence 3-hydroxyvalerate monomers makes PHB by V flexible and stronger. The properties of PHB by V are similar to those of polypropylene and therefore it is commercially more useful. Now let's know about polyhydroxybutyrate biosynthesis of PHB. Among the several PHA, the pathway for the biosynthesis of polyhydroxybutyrate, in short PHB, has been thoroughly investigated. Starting with acetyl coenzyme A, PHB is synthesized in 3 reaction steps. Acetyl coenzyme A is converted acetyl coenzyme A by the enzyme 3-ketothiolase, which is then reduced to 3-hydroxybutyryl coenzyme A by acetyl coenzyme A reductase. The reducing equivalents are supplied by NADPH. The enzyme PHA synthase is responsible for the addition of 3-hydroxybutyrate residues to the growing PHB chain. Now let's know about the regulation of PHB biosynthesis. The outline pathways concerned with the biosynthesis and degradation of PHB in Ralstonia eutropia we will now discuss. The enzymes of PHB biosynthesis are constitutive in nature. Thus, the enzyme machinery for PHB synthesis is present all the time in the cell. When the growth of the microorganism is restricted by limiting an essential nutrient, PHB synthesis rapidly occurs. During the active growth phase, acetyl coenzyme A gets oxidized through the Krebs cycle. Further, free coenzyme A inhibits 3-ketothiolase and therefore very little PHB is synthesized. When the growth ceases or is restricted by limiting a nutrient, the operation of Krebs cycle, that is oxidation of acetyl coenzyme A, decreases. This is mainly due to the inhibition of citrate synthase by NADPH. NADH. A decrease in the concentration of coenzyme A as citrate is not formed from acetyl coenzyme A. Coenzyme A is low, relieves the inhibition of 3-ketothiolase. This enables the diversion of acetyl coenzyme A for the production of PHB. The supply of reducing equivalents, that is NADPH, required by the enzyme acetoacetyl coenzyme A reductase also regulates the synthesis of PHB. PHB, the storage energy reserve compound, can be degraded into acetyl coenzyme A and metabolized by a Krebs cycle. This occurs when the organism is deprived of energy supplying carbon sources. PHB is degraded by PHB depolymerase to form 3-hydroxybutyrate. NADH is the inhibitor of the subsequent reaction catalyzed by the enzyme 3-hydroxybutyrate dehydrogenase, that is formation of acetoacetate from 3-hydroxybutyrate. The biosynthesis and breakdown of PHB from the cycling process we will now discuss. Now let's know about the production of PHB. Polyhydroxybutyrate is mostly manufactured by batch culture. PHB production occurs when there is an excess supply of carbon source and limitation of some other essential nutrients such as nitrogen, phosphorus or sulfur source. The production or accumulation of PHB is now we will discuss. There are two distinct phases, a growth phase and the polymer accumulation phase. As the growth phase ceases due to nutrient exhaustion, synthesis of polymer PHB commences. It is also possible to produce PHB by restricting the oxygen supply to aerobic bacteria. Now let's know about the applications of PHB. PHB can be implemented in the human body without rejection. This is because PHB does not produce any immune response and thus it is biocompatible. PHB has several medical applications, that is as durable bone implants for wound dressings. Attempts to use PHB as degradable sutures and other implants has not met with success due to very slow degradation of PHB. Now let's know about the biosynthesis of other PHA. Biosynthesis of other PHA is quite comparable to that described for PHB. Some important features with regard to synthesis of two important PHA we will now discuss. Now let's know about biosynthesis of PHB or V. Some strains of Ralstonia eutropha, that is formerly known as alkali genes eutrophos, are capable of synthesizing poly-PHB by V, that is hydroxybutyrate co-hydroxyvalerate. For the formation of PHB by V, glucose and propionic acid are required as substrates. Propionic acid as propionyl coenzyme A is responsible for the synthesis of 3-hydroxyvalerate. The three enzymes involved in the synthesis of 3-hydroxybutyrate 3-HB also participate in the formation of 3-hydroxyvalerate, that is 3-HB. The polymer PHB by V contains 3-HB and 3-HB monomers in a random sequence, that is no individual polymers of 3-HB or 3-HB exist. The relative concentration of glucose and propionic acid in the culture medium determine the chemical composition of pHb by V. Biosynthesis. Now let's know about biosynthesis of pHa with long side chains. It is generally observed that when a pHa producing organism is grown on a carbon source containing N carbons, 3 hydroxy monomers with N2 or N-2 or N+2 carbons are produced. This suggests the involvement of beta oxidation of fatty acids, which sequentially removes 2 carbon fragments in the biosynthesis of PHA. The organism Pseudomonas oleovarans can synthesize PHA when organic acids and alkanes are supplied in the medium as carbon sources. For instance, PHA with high content of 3-hydroxyalkanoate can be produced from N-octane or N-octanoic acid. Now let's know about Biopol A biodegradable plastic. Biopol was the trade name used by ICI plc in UK for the biodegradable plastics composed of PHB and PHB by V. The gram-negative soil bacterium Ralstonia eutropha was employed for the manufacture of biopol. Now let's know about production of biopol. The formation process for the production of biopol involves two distinct phases: biomass production phase or growth phase and polymer accumulation phase. This is comparable to the production of PHB. When the bacterium Ralstonia eutropha is cultured on a simple medium containing glucose and salts, growth phase is observed. By restricting the essential nutrient phosphate, growth phase can be stopped. Now glucose and propionic acid are continuously fed for large scale production of PHA, that is PHB or PHB by V. The relative concentration of glucose and propionic acid in the culture medium determines the proportion of 3-HB and 3-HB monomers in biopol. The biopol containing around 10% 3-HB is found to possess the desired characteristics. Now let's know about recovery of biopol. The cells can be disrupted and subjected to solubilization of components other than biopol. The polymer can be washed and recovered by centrifugation. Now let's know about limitations in the production of biopol. The manufacture of biopol by fermentation is very expensive when compared to the production of conventional non-biodegradable plastics. For this reason, many companies have stopped the commercial production of biopol by fermentation. Now let's know about the application of biopol. Biopol is approved for use as a food contact material. It is being used in the manufacture of paper cup and food trays. It is also used as a thin waterproof layer in food packaging. Biopol can be molded to produce bottles and several other items. Now let's know about the genetic engineering for PHA production. It is possible to commercially produce PHB by V and other polyhydroxyalkanoates by fermentation of R. eutropha. This organism, however, grows very slowly and utilizes only a limited number of carbon sources for growth. For these reasons, the production of PHA by R. eutropha is expensive. Fortunately, the genes responsible for the synthesis of important types of PHA by R. eutropha have been characterized and cloned. These genes have been transferred to E. coli. The resultant transformants, E. coli cells, grow rapidly and produce large quantities of PHB. But the major limitation is that about half of the E. coli cells lost the plasmid constructs in about 50 generations. This poses a big problem, particularly for large-scale continuous cultures. In recent years, some workers have tried to genetically manipulate and produce stable plasmids with some successes. Recombinant strains of E. coli have been used for production of PHB by V and other types of PHA. There are some other advantages of producing PHA by E. coli instead of R. eutropha. The polymers are produced in a crystalline state by E. coli, and their extraction and recovery are much easier. Now let's know about PHA production by plants. The major limitation of producing PHA by fermentation is the cost factor. Approximately it is 10 times more expensive to produce PHA by bacteria compared to the manufacture of petrochemical plastics. Plants are attractive sources for a less expensive production of PHA. A transgenic plant of Arabidopsis thaliana containing the bacterial genes for PHB genes was developed in 1992. However, the yield of PHB was rather low. Oilseed plants, which have plenty of acetyl coenzyme A, are attractive sources for PHA production. This is because the acetyl coenzyme A is the key substrate for PHA synthesis. No significant success has been achieved too far for PHA production by oilseed plants. Now let's know about the microbial lipids. Among the microorganisms, the eukaryotes can accumulate the triacyl glycerols. In contrast, prokaryotes predominantly accumulate polyhydroxyalkanoates. Now let's know about microorganisms as oil factories. In general, commercial oils used in foodstuffs and cosmetics are obtained from plants and animals. Thus, the traditional sources of oils and plants and animals. In recent years, microbial production of oils is gaining importance. There is a trend to use microorganisms as oil factories for some reasons. Let's know about these reasons. First is plant oils contain residues of pesticides and herbicides that are used in the cultivation of oilseed plants. Second is fats and fatty acid from animal sources are unacceptable by some sections of the society on moral and or religious grounds. Further, animal fats have risk of transfer of disease causing agents to humans. 3 is eukaryotic microorganisms are capable of producing a wide range of polyunsaturated fatty acids. Certain microorganisms can synthesize huge quantities of a single fatty acid, which is quite advantageous for large-scale production and easy recovery of a particular fatty acid. Fourth point is microbially produced oils, which are devoid of harmful contaminants, will be acceptable to the public on moral and ethical grounds also. Now let's know about the production of single cell oils. Oil rich in a particular fatty acids are regarded as single cell oils. So far, researchers have been successful in the producing single cell oils rich in 3 polyunsaturated fatty acids. These developments have occurred due to the interest taken by multinational pharmaceutical companies. Now let's know about linolenic acid. Single cell oil rich in linolenic acid was the first oil produced by the fungus Mucor carcinelloides. Its production, however, was later discontinued due to cost factors. Now let's know about the arachidonic acid. The soil fungus Mortilla alpina has been used for the production of oils rich in arachidonic acids. Now let's know about docosahexaenoic acid. The marine alga, this is the marine alga Crypthe, uh, Crypthecodinium cohni has been employed for commercial production of hexaenoic acid. Microbial DHA is used for inclusion in baby foods. Now let's know about limitations in the production of single cell oils. The fermentation technology used for the production of single cell oil is very expensive. It is visualized that the day may not be far off to transfer the genes producing single cell oils from microorganisms to plants. These transgenic plants can produce large quantities of single cell oils more cheaply. But these oils will be genetically engineered and therefore it is doubtful whether they will have widespread public acceptance. Now let's know about the microbial rubber. Natural rubber is produced by a large number of plants. Chemically, it is a biopolymer composed of cis-1,4 polyisoprene. The biosynthetic pathway for rubber has been worked out. About 17 enzymatic reactions occur for the formation of rubber. The substrates are simpler sugars. The final step involves the polymerization of isopentenyl pyrophosphate onto allylic pyrophosphate, catalyzed by the enzyme rubber polymerase. Attempts are on to synthesize rubber in genetically engineered microorganisms. For this purpose, the rubber producing plant Heavy brasilinensis was selected and cDNA library by using mRNA was constructed. constructed. It may be possible in near future to synthesize rubber from genetically manufactured microorganisms. Now let's know about microbial adhesive biopolymer. The biopolymer adhesive protein produced by the blue metal Mytilis edulis is very strong and waterproof. It is useful for the mucels to attach itself to different surfaces. As the adhesive is secreted, it gets randomly cross-linked, hence it cannot be sequenced. Fortunately, the immediate precursor for the formation of adhesive protein has been isolated and characterized. It is a 130 kilodalton protein rich in certain amino acids like serine, threonine, tyrosine, lysine, and proline. Most of the proline and tyrosine residues are hydroxylated. Further biochemical analysis revealed that this precursor protein contains repeating units of a D decapeptide peptide. The cDNA for the precursor adhesive protein has been constructed by using mRNA. This cDNA was introduced into yeast cells and adhesive protein molecules were synthesized by the recombinant yeast. The gene encoding the repeating unit of decapeptide has been identified and chemically synthesized. This gene was incorporated into E. coli cells for synthesizing adhesive protein. But there is a limitation. E. coli cannot carry out post-translational reactions, that is hydroxylation of the adhesive protein. This deficiency can be overcome by carrying out in vitro hydroxylation. It is possible to hydroxylate amino acids by employing bacterial enzymes in the presence of vitamin C. It is expected that the adhesive biopolymers will be soon produced for widespread use in dentistry and medicine. Now let's know about the microbial production of indigo. Indigo is a blue pigment extensively used to dye cotton and wool. There is a heavy demand for indigo world over, and it is at present the largest selling dye in the world. Originally, indigo was isolated from plants. Now it is being synthesized by using the hazardous compounds such as aniline, formaldehyde and cyanide. In the recent years, it has been possible to synthesize indigo from tryptophan by genetically engineered E. coli. The biosynthetic pathway for production of indigo we now discussed. However, a large-scale commercial manufacture of indigo by genetically engineered E. coli is yet to commence. Finally finished. Any questions regarding this topic, you can ask me in the comment box. I will definitely answer the question. And for more updates, you can subscribe my channel. Thank you.